Good morning. And what we have here, what I want to do is give you some notes. It's going to be a little complex. It might be a little long today. Fortunately, you have the class period to do this, so I'm not too concerned that it'll be long, but you need to make sure you get this done in class. It's due by the end of class today, so you want to make sure that you're paying attention, you're using your headphones, that you're doing it yourself, you're taking notes. And you're annotating those notes with any questions, okay? Same deal as always, it's just going to be a little longer because it's a little bit more complex because it's a little bit of biochemistry. We're going to talk a little bit about that today. So what we're really talking about here is molecules to metabolism. You should know that this is section 2.1 of 2.1 of your textbook. But we're going to a little bit more detail here, give you a little bit larger context here. But the big idea here is that living organisms, all things that are alive, all organisms, control their composition by a complex web of chemical reactions. It's really complex. So in fact, I want to give you a model of something here. This is a small subsection of part of the chemical pathways that happen inside of a eukaryotic cell. I think you'll recognize a couple of things here. We're looking at carbon dioxide fixation and ADPH and H+. We're talking about ATP synthase right here. So this represents a chloroplast doing photosynthesis, but we've also got some other chemicals that you might recognize. I saw pyruvate around earlier. I've lost it. Oh, here's pyruvate down here. We've got some ketones, which you might have heard of. We've got fatty acids, which we'll talk a little bit about today. We've got some ribose. We've got a bunch of oses here, a bunch of sugars. It's a really complicated process. And so this is what makes biology really difficult to understand, because we can understand these chemical pathways in isolation in a lab, but as soon as you introduce a chemical into these large, complex webs of metabolic reactions, it could have profound effects. It can conflate other reactions, and who knows the impact. So even though we do a certain kind of chemical reaction, a reduction or an oxidation or a phosphorylation, those processes don't always happen when the environment or other chemicals are present or not, because each chemical is going to influence the chemical environment that those reactions occur in, right? So it's very complex process. You do not need to know this. You don't need to copy this diagram down. I just want you to know that it's quite complex. So what we need to do, give you some context here, is, and know that biology has changed in recent decades. And pretty decades, maybe century or just over a century, but not quite so much in the public sphere, right? So there used to be this idea of stamp collecting in biology, which is really basically naturalists who would go out into the world on these adventures, often on boat, on ship, like Darwin did, collect specimens around the world, try to categorize them, classify them, right? Do you remember learning about Carl Linnaeus with his kingdom, phylum, class, order, family, genus, species categorizations? Just trying to categorize them, right? And because of that, biology has this reputation of being a soft science, as opposed to the hard sciences like chemistry and physics. And that's how a chemist or physicist would classify biology, as someone especially who has some old school training or doesn't understand the complexity of biology as we know it now, because there's this whole new field that we're spending quite a lot of time going into, and so we really make sure that we understand is that there's this idea called molecular biology that bridges the gap here between the quote-unquote soft science and the chemistry and physics. So there are these ideas that in biology that we can categorize living things based on the chemistry and the physics. And so to make sure you understand how these all work, physics will explain the chemistry, and then in chemistry, in some situations, will explain the biology. right? The physics and the chemistry will also kind of explain the geology, right? And that can help explain the biology. But geology and chemistry don't have to interact directly with biology, but they can, right? So these are all very much connected, and we've now figured out this connection pathway to biology, and with this idea of molecular biology. And there are two ways to look at biology, and depending on your perspective, there can be a reductionist point of view or a systemic point of view. And so the reductionist point of view is really kind of best exemplified with stuff that we've already done this year, right? So if you're thinking about the detail of the photosynthesis reaction or the cellular respiration reaction, those are both presented in a reductionist point of view, right? So we're trying to kind of distill these chemical reactions that chemical 1 is going to influence chemical 2, which influences chemical 3. But in reality, chemical 2 could have impacts on other chemicals that aren't in that specific pathway because they're all kind of interacting with each other inside the cell. Now the cell has a pretty good mechanism to prevent that when it's got some basic chemistry so that some chemicals don't interact well or easily. There are enzymes that help other things react, and then there are compartments inside of cells, those cell organelles. But another example that we're going to talk a little bit about is this process right here. So DNA being transcribed into RNA and then translated into proteins, right? So these processes are all, these arrows represent processes of, and you should have this transcription on this arrow and translation on that arrow. And it's moving in a single pathway, right? And we actually call this was a huge breakthrough in the 1930s, and we call this the central dogma of molecular biology, or biology. So a dogma is like a central core belief. We don't want to use the word belief in science because we have data for this, but this is a major tenet of biology. This connects the DNA to the protein, and it indicates that protein is not what's going to mutate DNA. So lots of things can happen to change this protein. You might also refer to this protein like as a phenotype, depending on what type of protein it is. But it's only when the DNA gets mutated that we would call it a genetic mutation. But there could be a problem here in making the RNA, or there could be a problem in making the protein, or really the next level, which is the folding of the protein. Right? There could be a problem there as well, and not be caused by a genetic mutation. So again, it's a lot more complex, and this is that systemic point of view, that one thing can influence many other things. Excuse me. Many other things, right? So you have to think of this as a large system and not reduce it down to one kind of reaction, okay? But we need to reduce it down real quick. So what we're talking about here is organic chemistry, right? So when we talk about organic chemistry, we need to be thinking about like carbon-based, right? Carbon-based organisms. And this is really where the term organism is coming from. So carbon-based organisms, or organisms are carbon-based, right? So carbon is going to be critically important, and yet carbon is not the most abundant element on the Earth. It's not actually near the top, right? So, but it's critically important, and one of the reasons it's important is because of its covalent bonds that it's able to form. So I need you to think back to chemistry, and I want you to think back to your molecular model of carbon, right? And so you remember the nucleus of an atom, which is different than the nucleus of a cell, has the protons and neutrons, right? So let's say we've got our nucleus here, and that carbon atom, if you remember, has 6 protons and 6 neutrons, has an atomic mass of 12, and it's got 6 electrons. Now, depending on which model you would resonate most with you, if you're thinking about these orbits, or maybe you learned about orbitals, either way, there are four valence electrons. Okay, so these four valence electrons are looking to create four bonds, four covalent bonds. And covalent bonds, because it's the sharing of those valence electrons, it can create four bonds. So if this is carbon, it can create four bonds. And those four bonds are all really strong. And what that means is that we can create these really complex chains or rings of carbon or carbon-based molecules, which gives us a wide variety of things. So carbon's a nice base for that. So, for example, there's a protein called titin. Okay? And titin is extremely, it's actually kind of complex in some ways and pretty simplistic in others. So it's the largest known protein. It is carbon-based. It's an organic molecule. And it has 539,000 atoms. All right, that's a lot of atoms. And its chemical formula is C169,723 H27046 N 45688 O 52243 and S 912. I might have copied one of those down incorrectly, but the point is it's a massive molecule, and the key here is this carbon, right? It's able to create those four hydrogen bonds, and that's going to be critically important. As a side note, biologists will color, and the reason this is all in gray is that carbon to a biologist is colored gray, but to a chemist we color black. It's all arbitrary anyways, because carbon itself as an atom doesn't have color to it. And so when we talk about these carbon-based molecules, this organic chemistry, we're going to break this down into the four categories that we've already mentioned related to your aquaponics project: carbohydrates, lipids, proteins, and nucleic acids. So nucleic acids. Okay, and that's really going to kind of be where we talk about this. So let's talk about carbs first. Alright, so carbohydrates. It's really important to make sure you understand the term that's going to help you. So this is carbon, and think of hydrate as in water. So we know that the atoms, we'll do that a little better, are carbon, hydrogen, and oxygen. And we've got H2O here, that's the water. So we know that for every one carbon, we have two hydrogens and one oxygen. But I'm going to give you a little caveat here. This is only true for monosaccharides. Alright? So monosaccharides, mono meaning one, sugar, saccharide meaning sugar, monosaccharides are the monomer of the carbohydrates. It's one of the kinds of monomers of all the organic molecules. And you want to make sure you clarify this term. Monomer is a generalized term. It just means one building block. And so when we take two monomers together, we make what's called a dimer, right? And we put multiple monosaccharides together, we get polymers. And so in this case, you can guess that two monosaccharides put together is called a disaccharide, and three or more is called a polysaccharide. And there are going to be some known examples that you need to know, and we'll talk a little bit more about this. But really what you want to know is that these carbohydrates are made out of one or more simple sugars, right? So a monosaccharide is a simple sugar. And so let's go, actually we'll go ahead and make our list. So I'm going to write mono, di, and polysaccharides, right? And so for our monosaccharides, there's a list that you need to know, and that you need to know. It's good to know this list. So we've got glucose as one example. You could have ribose. You could have deoxyribose. You could have fructose. For the disaccharides, lactose is another one you could have. You could have maltose and sucrose. And for our polysaccharides, the ones you need to know are cellulose, amylose, that's amylose, which is another name for starch, and glycogen. And you need to know that list, where they go, how they get categorized, because as we take these big molecules and break these down into smaller molecules, we call that reaction catabolism. And building them up out of simple things into big things is called anabolism. But this ratio of 1 to 2 to 1 is only true for this category right here for monosaccharides, because of what's happening in the anabolic reaction that removes some of these molecules. And you saw this already when we talked about ATP and making ADP into ATP. If you go back and look at your POGL, you'll see that water was removed when we combine molecules. And then what you should know is that commonly these carbohydrates follow this kind of ring structure. So sometimes they're drawn like this. So it's a ring, and anytime there's a bend or a corner, that actually represents a carbon, usually. But this would actually be an O, an oxygen there, and you might have a carbon there, right? So that carbon looks like it only has one bond. This one has 3 bonds. This one only has 2 bonds. So really what we see happening, I'm going to redraw this with a little bit more detail here. So we have a bond here. I'm going to put my oxygen molecule there. I've got this C, and it's got 2 hydrogens that we don't show in this model. This one has 2 hydrogens. It's actually, because of the formula, let me write it the right way. It's a COH. This is going to be an H and an OH. Another C, an H, an OH, C, OH on the inside, H on the outside, C, we'll put an H here. And what's interesting is that carbon now has its 4 bonds, but this one, let's count up, well, this is going to have 2, 3 hydrogens on it. Let's count up our oxygens. 1, 2, 3, 4, 5. So this actually should be an OH. So I get my 6 oxygens for my 6 carbons. 1, 2, 3, 4, 5, 6. And as a separate note, I'm going to tell you that we number these carbons. So when it's drawn like this, here's my oxygen. So the first carbon to the right of it is 1. This is carbon 2, carbon 3, carbon number 4, carbon number 5, carbon number 6. So this happens to be C6H12O6, which we know as glucose, but that's an isomer. It's the same chemical formula here as fructose, in which this molecular structure looks different. Now that looks messy, so there's yet another way to write it, and I'm going to draw that up here. So we could do CH2, sorry, it's not CH2, it's COH2, right? Does that make sense? And you could abbreviate it that way. and you could have CH3 up here, right? So I think you can figure out the rest of them there. Or even more simple than that, we can draw it. Instead of drawing all those letters, put an O there, you draw a line like that, and you've got your ring. And anytime there's a bend, again, there's implied a carbon, but because we know that this is a carbon, there are two bonds, the other two must be H's, and in this case, OH's. So our carbohydrates are rings like this, or they might look like I'm going to draw one with one less carbon, right? So it could be these long chains of more rings that continue down, and we get our polysaccharides, which just two would be a disaccharide. All right, so you should be able to recognize those. There's some pictures in your book that you should reference. Let's talk about lipids. All right, so let's talk about lipids. And this is a weird group, but anything that's considered a lipid has to meet this one characteristic, that it's hydrophobic. All right, and in chemistry, you use this term nonpolar. Right, so we'll talk a little bit more about this when we talk about the properties of water. But you need to know that these lipids all of them have carbon, hydrogen, oxygen, and sometimes some of them have a phosphate group to them. So some of them have phosphorus. Now they all have a hydrophobic component, but there are three major categories that you need to know. You need to know fat as one type of lipid. You need to know phospholipids, which happen to have a hydrophobic part and a hydrophilic part, and then steroids. But they all fit this category of hydrophobic and nonpolar things. And you might have heard of this these fats as triglycerides, and there are two kinds of simple lipids and oils. And so fat is that's solid at room temperature, and oils are liquid, that's saturated and unsaturated. So if you don't know what that means, I need you to ask me in class on Friday. But I want to give you the generalized structure of a fat, right? So this is a fat molecule. I'm going to color code this into different sections. So there's a section that's a 3-carbon glycerol. So it's got 3 carbons, but it's also got some hydrogens on here. And this is actually a really big molecule. Okay. So this part that's in orange is a glycerol. And then I'm going to do another part that is a part of the fatty acid tail, right? So it's a long hydrocarbon chain. So it could be really long. We could keep going. I'm going to stop there. And so it's a hydrocarbon chain. So we're going to have a bunch of hydrogens that come off of each of these carbons to give it its 4 covalent bonds. I'm going to write the next one similarly, but with a different formula, so you get accustomed to it. So this is going to have that same base here, but I'm going to write it a little differently. So I've got 1, 2, 3, 4, 5, 6, 7 carbons in this chain, and 6 of them have 2 hydrogens to them. So I'm going to write it like this: CH2 6 times, and then a CH3 on the end. This is my CH3. So I could write it that way. And it means the same thing. No, these fatty acids don't have to be the same length. But I'm going to draw, so these are fatty acids. And because these fatty acids have the maximum number of hydrogens, we call this a saturated fatty acid. And if we had 3 of those saturated fatty acids on each of these carbons, we'd have a saturated fat. And saturated fats are solid at room temperature because they're these straightish lines that you can pack in real tight. But I'm going to draw an unsaturated one here. So I'm going to make this one, let's say, a little shorter. And we'll make that a CH3, just like over here. But let's say we've got a double bond, a double bonded carbon here. That actually bends the structure of this tail, so it actually would be kinked out this way, or maybe this way. And I still have 2 hydrogens here, but I already have 1 bond, 2, 3 bonds, so I'm only going to have 1 hydrogen over here, and I'm going to have another hydrogen there. That's still my 4 bonds, and this one is going to have 2 hydrogens. Right? So this is an unsaturated fat. Now, an unsaturated fat is liquid at room temperature, and that has some serious implications, solid versus liquid or oil, towards your health and your cardiovascular health. And when they're on, these hydrogens are on the same side, so notice this hydrogen and this hydrogen on the same side, it's known as a cis unsaturated fat. But oftentimes we don't have cis unsaturated fats. Our food has this other kind of fat that you've heard of called a trans fat. And trans, meaning across, indicates that this hydrogen is simply on the other side. It's a cross, and that would be called a trans unsaturated fat, fatty acid. And that's what you need to make sure you understand and can draw and can identify as far as the lipids, as far as a fatty acid goes. What I'm going to do now is kind of change this around a little bit so that we can get a phospholipid bilayer. So what I'm going to, let me just bear with me here. I'm going to erase, so I'm now going to be drawing a phospholipid. Which will be similar. I still have my glycerol. I've got 2 fatty acids. And we end up with a phosphate group on this end, but remember phosphate groups have a negative charge on them. It's actually 3 negative charges. So the negative charge is now the this is the phospholipid up here. A hydrophilic charge, because it's going to bond with the positively charged hydrogens of the water molecule. And that's our phospholipid. It's got 2 fatty acid tails as opposed to 3. Steroids are several rings that are also hydrophobic, and so they often find themselves buried in the hydrophobic section of the phospholipid bilayer of cell membranes, and they control development of development of your body and different developmental stages through your lifetime. All right, so I'm going to stop at one more category for now. As I say, I'm already at 30 minutes, so I want to talk about proteins, and I think that's where I'm going to stop. And I'm going to ask you to do a little bit of reading in your textbook for homework. So for proteins, we've already talked about this, that they have these molecules in them, carbon, hydrogen, oxygen, and nitrogen, and sometimes they have this molecule of sulfur. Not always, but sometimes they have sulfur. Some of them do. And these things are made out of amino acids. So you need to know how to draw an amino acid, and so I'm going to show you how to do that. So I'm going to draw it in gray first, and then we'll relabel the parts that you need to be familiar with. So what you need to be familiar with is the first component, which is an NCC backbone. It is the cornerstone of the amino acid, and then there are two hydrogens that are bonded to that nitrogen. This carbon right here is called the alpha carbon, but it's just the same kind of carbon, but it has a very important role. We have a double bonded oxygen to that carbon and an OH. This carbon right now only has two bonds, so one of those is going to be hydrogen, and the other one is something called an R group. What we do now is kind of color code the different components to it. So what we have our backbone, right? Our NCC backbone. We always write it in this sequence, NCC. We have our amine group with the nitrogen. That's my amine group. And then I have a carboxyl group over here. A carboxyl group. So we've got an NCC backbone amine group, a carboxyl group, which is where the amino or carboxylic acid group. And so the amino acid name comes from that. But then there's this R group, which is not part of your periodic table. This stands for residue group. That's where the R comes from, but sometimes it's called a side chain. And that R group is the variable thing. Sometimes it's called a variable group. This is what makes each amino acid different. There are a total of 20 amino acids that help you function, that keep you alive. It's kind of like letters of the alphabet. We can put them together in different ways with different repetitions, different lengths, and make different words. We have 20 amino acids to work with to make all the thousands of proteins that keep you alive, that range from, and I want you to write this list down, hormones like insulin, enzymes like catalase or our NADPH reductase or ATP synthase, transporting gas around your body, not methane, but like oxygen and carbon dioxide, or immunoglobulins like antibodies that are made by beta lymphocytes and helper T cells. So they do lots and lots of things for us, and this is the basic structure, and you need to know how to recognize that. I'm going to skip over the nucleic acids, but I do want to show you just a quick, I want to show you two amino acids that that could be helpful for you to know. Actually, maybe three real quick. So what I'm going to do is just put them down here on the bottom. So I'm going to give you the simplest one first. I think I want to give you the simplest one. Yeah, I'll give you glycine. You don't need to know these, but I want you to recognize the similarity in them. So we have our amine group, our NCC backbone, and our carboxyl group. And this right here, that hydrogen, is my R group. In this case, it's just a hydrogen. Here, we have a CH3 as the R group. We could, and that's alanine. But you could have a larger one that's like, I'll give you arginine. And I'm going to draw it a little differently because I'm running out of space upward. But it doesn't matter how you draw this. So this alpha carbon, I'm going to put a hydrogen up there, but the R group is really coming down in this case. It's going to be CH2, a CH2, a CH2, a nitrogen with a hydrogen attached to it, to a carbon, double bonded to an NH, and an NH2. And that R group is far more complex than these. And this, because they have different chemical properties, and they can combine in different ways, they'll actually take up three-dimensional space in different ways as well. So you need to make sure you understand those components and be able to recognize those. But what you need to also, what I want you to prep and further read is the section on anabolism and catabolism, and make sure you understand what's happening to the substrates or the reactants and the role of water, whether water is being used to break something or water is being created through the reaction. And you need to get those two terms straight, because we're going to add some more complexity with these structures and those terms. Because those terms can be applied to all of these structures. On Friday, you need to be prepared to understand this, to show me that you understand it. You also, you may see questions. You'll check Google Classroom. You might see some questions posted in Google Classroom that I want you to answer in class now. And I want you to be ready to talk about and share the question that you are investigating for your science fair projects on Friday to the class, and to give a status update about where you are in that process, whether you just came up with the idea, you're still coming up with an idea, which is a terrible place to be, but that might be where you are, and you need to figure that out as soon as possible. If you've collected data, if you've started collecting data, or what your plan is to collect data, because there are only a few lab days left. Because your labs, your in-class science fair before the school science fair is going to be on January 19th. So your in-class fair is January 19th. The last day for data collection in my lab is January 13th. That does not leave you a lot of time to finish your report, make your board, and present it to the other science teachers that are coming into my room to make the call to see if you are going to be able to join us on the 28th for the school science fair, which would make you eligible for the regional science fair. Okay, I think that's long enough for you. I'm at pushing on 38 minutes here.